Okay, so the last part of this chapter, proteins gone wild. So one is called racemization. So in long-lived proteins like collagen, elastin, some of the L amino acids can actually convert into D by just random racemization. It happens every so often. And then arginine can be oxidized to citrulline. Citrulline containing proteins are serological markers for chronic inflammatory diseases such as arthritis. So that's the problem with having proteins that try to last forever. Racemization and arginine oxidation, among others. And then sometimes you can get incorporation of the wrong amino acid into proteins. So in chapter 33, we'll talk about sickle cell disease, BMAA in chapter 36. And then we have glycation, or non-enzymatic glycation. That's when reducing sugars, now you know what reducing sugars are, form bonds with lipids and proteins in an uncontrolled manner at high concentrations of reactive sugars. Glycosylation is an enzymatically controlled system. So glycoproteins formed by glycosylation are natural. Glycoproteins formed from glycation are not natural and have pathogenic properties. So here's a chemical equilibrium. Okay, this will happen. Under normal conditions of glucose concentration, it doesn't happen to any significance. But what if you have constant high amounts of glucose? By Le Chatelier's principle, you increase one of the reactants, you expect to get more products. And that's a problem that we'll look at in, I think, chapter 33. And then chaperones and the ubiquitin proteasome pathway. So chaperones are proteins that help other proteins fold correctly as they're being synthesized by ribosomes. They prevent misfolding and aggregation of existing proteins. They use ATP. They help proteolysis of misfolded proteins. And once a protein has become misfolded, it's got three possible fates. It can bind to a chaperone and either be refolded back into the native state or remain misfolded. It can form an aggregation with another misfolded protein, or it can be hydrolyzed by proteolytic enzymes if it's unfolded. So chaperones were first discovered as a class of heat shock proteins. So in particular, they discovered it in plants. Plants have a way to protect themselves from being overheated. So when plants or any organism, when they're exposed to high temperature, genes are activated that code for chaperones, and the chaperones are there to prevent proteins from being misfolded. And if they are misfolded, to turn them back into correctly folded proteins. So here's what can happen with a ubiquitin proteasome pathway. So ubiquitin marks another protein for death by another system, which is called a proteasome. So what does that mean? So ubiquitin was a protein that was ubiquitous. They were finding it everywhere. It's like, what is this protein? It's being bound in multiple copies oftentimes to other proteins. What's the deal with that? Well, what they discovered was that you get these misfolded proteins, ATP is required to put ubiquitin onto the misfolded protein. That marks it for death by what's called the proteasome. So while ubiquitin is a small protein, the proteasome is huge. So here's the ubiquitin binding site on the proteasome. So it grabs this, and then it just simply degrades the protein. So this is one great big giant, how do you say, proteolytic enzyme.
So is ubiquitin a type of chaperone?
No, it just seems to be a marking protein. Like here's these misfolded proteins. They're not getting chewed up by existing proteolytic enzymes. So you mark it with ubiquitin, and then it can bind to this big proteasome right here.
So where is the chaperone in all of that? Is that part of the proteasome?
No, the chaperone tries to fix it. But if it can't get fixed, then it's either going to be cut up by proteolytic enzymes, or it goes into the ubiquitin proteasome pathway. So anyway, when the proteolytic pathway is not, how do you say, working correctly, like especially with age, it starts to become less efficient. You get an accumulation of proteins that are misfolded. You get these aggregates, underlying cause of many neurodegenerative diseases. I've never really heard it talked about in terms of Alzheimer's and stuff, but I would suspect it has something to do with it. So here's just more of a sequence over here. And essentially you simply break it up into small little pieces, right? So this is simply a more information-filled one than this one. So here we're just calling it the system. Here is the system. Okay. Then another one that's related to this are prions. So prions cause mad cow disease, other brain diseases. And here's the way I wrote it over here. I think this explains it quite well. So there's a prion protein. The SC stands for scapi disease. It was originally found as the brain disease in sheep. And anyway, it's able to take a regular prion protein, which has a function, and turn it into a bad one. So here you have mostly alpha structure. Now you get a whole bunch of beta structure involved. And that's why these are called amyloid beta fibers sometimes. These form plaques. And so what is it that turns a normal prion protein into the disease one? It is the disease one itself. So the scapi prion protein is acting as a chaperone, but in a bad way. It misfolds the regular prion protein into a pathogenic problem. So here you have a protein causing another protein to go bad. So this is not a very good picture, but I don't have a lot of good pictures for this. So here's the infection, right? So here's your regular prion protein. It comes in contact with the SCAPE protein. It acts as a chaperone, and it starts this vicious cycle. So it takes the regular one, converts it into a prion one, and simply starts forming these plaques. These are the plaques that are being formed. So this guy, Stanley Prusiner, is kind of like a hero to me in a way. So he was the main proponent of the prion-generated disease theory. A disease caused by a protein. There's no bacteria, there's no virus, there's no DNA. He was ridiculed for this. It's like, can you imagine going to some conference and you're trying to tell everybody in the field that no, there's no bacteria, there's no virus, there's no DNA. A protein caused this disease. Well, eventually you got the Nobel Prize for it. So another prion called alpha-synuclein was formed in 2015, named in 2015. It seems to be associated with Parkinson's and another disease. And the same protein had been studied in Parkinson's, shown that it could generate protein misfolding, but it's never shown to cause the neurodegenerative disease. And so Prusiner was still working on prions, even though he got the Nobel Prize. Some people just quit. Hey, I made it, you know? So he engineered a strain of mice that produced the human version of alpha-synucleins. And brain samples from diseased patients were homogenized, placed into the brains of genetically modified mice, then got the disease and died. So there was misfolded alpha-synuclein. So when brain samples from deceased Parkinson's patients were placed into the same genetically engineered mice, there was no aggregations of misfolded alpha-synuclein, even though the same protein is involved in both diseases. So it's not quite as simple as this, but that's the general theme of things. So amyloidosis or beta fibrillosis results from plaque forming neurodegenerative diseases such as Alzheimer's and ALS. And so amyloids are aggregates of twisted beta-pleated sheets that come from regular proteins that have undergone partial hydrolysis. So with partial hydrolysis, that is incomplete destruction of the protein, some of the alpha structures convert into beta structure that then causes beta amyloids. Okay. Now these become rather inert to proteolysis, inert to being removed by phagocytosis, interaction with antibodies, and yeah, it's a result with problems with the chaperone, ubiquitin proteasome pathway, or simply with infection from prion proteins. Right now, it's the only leading cause of death with no known cure, no preventative treatment, or any way to slow down its progression. So here's one insight into this. Presentilin turns into beta-pleated sheets that then form these amyloid plaques. So here's what we know about it. So here's a regular protein, okay? It's a membrane protein. And then when it gets degraded So here's being degraded. That's supposed to be destroyed. That's degraded. Supposed to be destroyed, right? That's supposed to be destroyed. But this part right here, for some reason, starts to accumulate and turns from alpha structure into beta structure. So it's a problem when you try to recycle these proteins that you get this amyloid plaque forming. So going back to this system, right? Maybe it's a problem with ubiquitination system. And you can see it gets rather complex here. So if anything goes wrong or doesn't work quite as well, then you're not going to get these proteins degraded. And the reason why you want them degraded is because you don't want a bunch of proteins hanging around without a purpose, because this kind of stuff can happen. So anyway, I haven't heard much more about this, and I didn't put a date on here, which I should have. I should have put the publication where I found this on there. But look at this structure right here. It's called monophospholipid A. And this has been shown, at least in mice, it reversed some of the symptoms of Alzheimer's. So look at this molecule over here. So here's, it's like aminoglucosamine. It's phosphate on here, so it's phosphorylated over here. There's carboxylic acid over here, another carboxylic acid, a bunch of alcohols over here. So all this part right here is pretty polar. If you look down here, here's a fatty acid. Here's another fatty acid. This one is, let's see, alpha beta hydroxy. Here's another one. So all this stuff over here is nonpolar. So it looks like an emulsifier. It looks like it would act as a soap. So if this is able to dissolve these plaques, then at least in mice, then that's promising. Who knows? So your immune system tries to protect you from bad proteins. And so I just generally put this in here. I don't expect you to know everything that's in here about what type of blood cells there are and all that kind of stuff. But white blood cells, or the lymphocytes, include B cells that release antibodies, natural killer cells that kill tumor cells and cells infected with viruses, and T cells. So you have T helper cells, cytotoxic T cells, you got regulatory T cells, and we'll be talking about the regulatory T cells later on, because these are supposed to suppress the immune response after an infection and prevent autoimmunity. Anyway, let's see, what else do we want to say about that? So when regulatory T cells don't do their job, overreactive cytotoxic T cells can cause significant damage to normal cells. So this is what happens with allergies and type 2 immunities. Okay? So the antigen-antibody complex causes mast cells to produce histamines, prostaglandins, leukotrienes, resulting in all the symptoms, right? And so that's part of the problem with allergies. When your cells right here don't work the way they're supposed to work. So anyway, allergic response reactions may be nature's way of keeping us away from harmful antigens and environmental conditions. But it's kind of can be deadly. Well, here's where I was talking about that. This probably should be in chapter 36, maybe not so much here. So here's what normally happens. Somebody hears about peanut allergies killing people, and so they set out to protect their babies from peanuts. And that's probably the worst thing they could do. So there's a lot of evidence that you feed babies peanuts at low amounts, they will get an immunity to it. Okay? And that's what they're finding. And a lot of this stuff the concepts came from looking at two groups of people who don't really mingle too much with regular society, the Amish and the Hutterites. And when they looked at these two groups of people, one group came in contact with animals a lot and the other ones did not. So the children who did come into contact with farm animals were coming down with all these allergies and everything else. Now you realize that these groups of people aren't eating the same foods that we are. That's why they were looked at. So now they recommend that children that don't come in contact with farm animals should be exposed to the dust from these farm animals so they can create antibodies against the microbes. So they're thinking that would help prevent the development of asthma. So here's kind of an off-the-wall experiment. Now they're talking about, you've heard about bees being depleted, right? People worry about bees a lot because bees are responsible for so many of our crops and everything. So they're finding out that vaccination can even be used with invertebrates. So pretty interesting experiments going on, okay? So the queen bee can actually pass disease-causing bacteria and hopefully cause immunity for the other offspring. That's what they're going to be trying now. And celiac disease and gluten. So gluten is a composite of several different proteins commonly found in these foods, okay? And anyway, here's the problem. So it's made up of gliadin, glutenin, and gluten. So this is soluble in ethanol, not very soluble in water. This is soluble in acid or in base, and this one is even harder because of its a combination. Okay. So a recent paper listed 55 diseases that could be caused by eating gluten. So here's the problem. Gluten gets only partially digested in the stomach. Then when it enters the intestines, it can permeate the intestinal walls and enter the surrounding tissues. So here's a blow-up of this. So people who are gluten sensitive and have celiac disease and all this kind of stuff, you see these villi over here? That's normal. But look what's happening over here. They're just disappearing because the T cells are attacking them. That's why. So here's the gluten proteins. They permeated the wall of the intestine, which is not a good thing at all. If you have a leaky intestine, that means that these gap junctions in here are letting things through here. that's a problem. Leaky gut syndrome causes inflammation and all kinds of other problems. And what does gluten do? It's a similar thing. So it can cause leaky gut syndrome. But in addition to that, this partially digested protein that can't be digested further causes an immune response. This is an antigen presenting lymphocyte. So what it does is it grabs on here and it says, oh my God, here's a protein that's bad. And we have to make antibodies to attack it. And you see the T cells are producing cytokines and everything else. It's just an inflammatory war over here. It's just incredible. And that ends up leaky gut syndrome, damaging all the microvilli on the cells right here, greatly decreases your ability to properly get nutrition. And even worse, this inflammation stuff causes havoc all over the rest of the person's body. So really, the body's reaction is really an autoimmune disease. Let's see, what else can we say about that?
So would you say eating gluten is bad for everybody, or just if you're gluten intolerant or have like celiac?
Yeah, if you're gluten intolerant, I think that's the issue. And so here's the problem. Humans have not adapted to gluten. So why is gluten even there? Well, many plants have toxins and different ways to protect themselves from being eaten. And gluten appears to be one of those. So they put gluten in the husk to protect against predators. Now, birds have been eating them for millions of years, right? So any bird that couldn't handle gluten died, and the only ones that are around are the ones that can handle gluten. But most mammals have not adapted to grains and do not eat them in substantial quantities. That includes humans. And so when we were hunters and gatherers cruising around, nobody's going to sit there and try to get a whole bunch of grain from a wheat plant or something. Wheat did not grow in big fields with people cultivating it. Nobody baked bread and stuff out of this stuff. It was only after farming and cities and the ability to make bread and stuff like that, that people started getting exposed to large quantities of gluten. If you go back to the Middle Ages and when all this cities and stuff were happening, one of the major causes of death back then was simply, what was the word for it? God, it was just right on the tip of my tongue. Where you just waste away. They didn't have a cause for it, but people just wasted away and they died. What is the word? God, I can't think of that right now. Consumption, that's what it was. People died from consumption. So what that means is that the people who could not tolerate gluten, they simply died and nobody knows why they died. So those populations that survived it were the ones that could survive gluten in the diet at higher concentrations. Now people are traveling all over the world. They're eating all kinds of different foods. You go into a Safeway and look at the foods there, you don't even know where all those parts came from. It's a worldwide food supply. So some populations basically suffered the consequences of gluten and much of their population died out. And other populations of the planet never went through that. So now they're having problems with it now. It seems to be that moderation doesn't solve the problem. Even a small amount can be terrible because there's an immune response. So there should be gluten-free diets. So if you are gluten tolerant, should you go out and be on a gluten-free diet? And I would say no, probably because you're not going to get as good of a nutrition as you would otherwise. But people who are gluten intolerant should be diagnosed and they should have gluten-free diets. And anyway, so there's a molecule to replace gluten. So you can make bread and things like that. It's called Wellens. It's made by Dow Chemical Company. And anyway, I don't know if it's available to the general public. It is available to commercial bakers and stuff. You can buy gluten-free foods, right? Anyway. Another thing that we have to think about is another molecule called fructans. So people probably have an issue from the fructans, and then they think it's celiac disease, so they start to avoid gluten. And yeah, I'm not so sure that's the best thing to do. We just need to have better ways to diagnose what's going on. Oh yeah. Yeah, this is actually kind of interesting. This protein on the egg membrane that was called Juno, it fuses with its counterpart on the sperm molecule called Izumo. And the interaction there was so weak that they could not find the mechanism how a sperm and an egg actually combined. They could not find what was really causing it until 2014. So they had to go a roundabout way to get it, to figure out what it was, but they did it. Pretty interesting. So that completes this chapter. Anybody have any questions about anything in this chapter? So you should know the basic structure of an amino acid. You should be able to take any of the common amino acids and put them into one of these categories. You should know what electrophoresis and isoelectric focusing are. And levels of protein structure. And you should be able to draw the amide bond between amino acids, the peptide bond. So if I gave you the structure for alanine and serine, you could draw the complete structure of serinoalanine. And then I gave a lot of different protein examples and how we categorize them and stuff like that. This is also important, the 5 major forces that stabilize protein structure. Let's see. Any questions on anything as we pass through there? What cooperativity is? Let's see.
Can you go over the different classes of immunoglobulins again? That chart right there?
Sure. Yeah.
I don't really understand what the chart is saying.
Oh, well, they're just trying to categorize them as to where you find them, what concentration they are, and what their functions are. And so if you come across this structure, right, it could be IgG, IgE, IgD, IgA. So those are all possibilities for those, but then their functions are different. Where you find them may be different, and their functions are different. And so, yeah, IgG is the most prevalent one in the serum. IgA is the most prevalent one in secretions. IgM, which is the big weird one, is also in the serum. IgD is found associated with membranes, as is IgE. IgE is mostly associated with allergies as well. Is that what you mean? I mean, they're just trying to find a way to, hey, here's what everything is. It's not like, oh, because of this, then this. It's simply a presentations of the types and how much there are and what forms they come in.
So serum is blood, and is secretions like your saliva? Like,
And the
okay.
secretions that protect, say, your lungs and other organs and things like that.
Okay.
Mm-hmm. So let's see. And hopefully you know what the complement receptor is, the FC portion. Where is that? That's here, right? And there is this used in multiple things, including complement. Let's see. So we're going back to proteins gone wild and incomplete digestion. You can see why there's a problem with the proteolytic enzyme, right? If part of it is folded up in a certain way, the proteolytic enzyme can't even get a hold of it. So if it doesn't denature in the correct manner, then yeah, you can't get a hold of it. And say this was all folded up over here and this part was not folded up. Okay, so you chew off that part right there, and now you still can't get a hold of this. So it can become a problem. I think one of the things about the proteasome pathway is that if you have a protein that can't be attacked simply by the proteolytic enzymes cruising around, marking it for death with ubiquitin is probably a way to pull it onto this big structure over here and forcibly denature it. But again, the ubiquitination pathway is pretty complex. A lot of different things involved. Okay, so any other questions then? We can review this later, of course. Okay, well, we'll start the next chapter then. Which is enzymes. I'm multitasking here. I have to keep checking my email all the time too. So enzymes are pretty much all over the place. They do things we don't even think about. So when you crush garlic, that activates an enzyme called allanase. That converts the amino acid cysteine into sulfenic acid named allicin. So this is what is the smell of garlic. Then you can reduce the garlic odor by using lemon juice. So what does that do? It denatures allanase. So it prevents the formation of the allicin. So timing and everything else is really important in cooking, but it's really because of you're controlling the enzymes. Fruits that brown when exposed to the air, like apples, contain oxidases. They can oxidize many of these odorous compounds. Anyway, there's this food and digestion, everything else. It's just, we don't even think about it, but it's all enzymes. So an enzyme can easily catalyze a reaction, speed it up by a factor of 1 billion without changing the temperature, without changing the pH. And they're very specific. Most of them will react on only one substrate. And we use the word substrate in term instead of reactant. And there's no byproducts. We just got through looking at what happens when you have incomplete digestion of a protein. That's a byproduct. It's not taking care of it. It causes havoc. So most of the enzymes are stereospecific. And we simply can't afford as a cell or as an organism to have unwanted byproducts. Let's see. So most enzymes are conjugated proteins. So the holozyme is the entire biologically active or native enzyme. And you can take the holozyme and separate it into what's called the apoenzyme and the cofactor. So the apoenzyme is simply the protein part, usually not active. The cofactor is the non-protein part, also usually not active. And the cofactor could be either a coenzyme, which is an organic molecule non-covalently bound to the protein. It's usually, and not usually, but often, a water-soluble vitamin. And what the coenzyme does, it accepts or holds on to a functional group during catalysis. And then the cofactor could also be a prosthetic group, which is like a coenzyme, except that it's covalently bonded to the protein. And then it could be a metal ion, which is a mineral that holds on to electrons during catalysis or forms coordinate covalent bonds with some functional group. So if you think about what we need in our diet besides nutritional things, which would be protein, carbohydrate, and lipid, we need vitamins and we need minerals. Why do we need vitamins and minerals? Well, because they're cofactors for enzymes. So here's something that really blows my mind to this day. If you look at all the things that enzymes do, all the things that cells can do because of those enzymes, there's only 6 classes of enzymes. Wow, that's crazy. So the forward and reverse reaction are in the same class, okay? Most reactions are reversible. And remember, a catalyst does not change the point of equilibrium. It doesn't change that at all. All it does is make you go to equilibrium faster. whether you're starting from this way or with this way. Doesn't matter. So the first class is the oxidoreductases. They perform redox reactions using NAD, NADP, FAD, FMN, or iron. And remember, in the organic chemistry part of this class, we were talking about these as either X or XH2. So here's XH2, here's X. Get it? So here's an example, alcohol dehydrogenase. This enzyme in your blood takes ethanol, which you put there for one reason or another, and it takes X and it oxidizes the primary alcohol into an aldehyde, and now you have XH2. Okay. Here's another one, pyruvate, the end product of glycolysis. What is this right here? This is XH2. So I'm going to reduce pyruvate, and I get lactic acid. And you guys know why you produce lactic acid. Later on, we'll see that you make lactic acid in response to anaerobic activity. But really the reason why you make lactic acid is for one reason, and that is to regenerate X. If you don't have NAD, all the other processes that are used to make ATP comes to a halt. So you need NAD. That's why you make lactic acid. Class 2 are the transferases. So transfer is a group of atoms from one molecule to another. Sometimes we call it a functional group that gets transferred. So here's ATP plus creatine to give you ADP plus creatine phosphate. What happened here? We took the phosphate and transferred it to creatine. It's a transferase. Okay. So a creatine kinase is found in muscle tissue, and the purpose of it is that you can store high energy phosphoanhydride bonds in the form of creatine phosphate. So in case you ever need more ATP, this is a reserve of ATP because the phosphate can be transferred back to ADP to make ATP again. It's just going to be used for muscle activity and nerve activity. And the other one I have down here is DNA methyltransferase. The transferase that methylates DNA and deactivates genes in the regulation process called epigenetics. So here's a base in DNA. So in DNA, you have G, C, A, and T. So here would be the C. So here's the donor, and there's the methyl group. You see that sulfide right there? Maybe this is a reason why I should add sulfides to the conversation in that part of the course. That's the methyl group. So right now, that sulfide is loaded up with this methyl group right there. If I didn't have the methyl group on there, sulfur would have two bonds, and it's simply sulfide. So there's an enzyme that takes this SAS, puts the methyl group on it. And then another enzyme comes along and takes this, and takes that methyl group and transfers it right there. So this is 5-methyl cytosine. This is the methylated base that you see in DNA. And then we have hydrolases. So hydrolases break bonds using water. So glycerol, triacylglycerol being hydrolyzed into glycerol and 3 fatty acids. That's the lipase of adipose tissue. Here's ADP plus phosphate making ATP. So you're basically hydrolyzing in the opposite direction. So remember what I was saying about the categories? They're both directions in the same class. Does that make sense? Obviously, that's hydrolysis this way. But what about when you make ATP? The ATP synthetase of mitochondria, that's also a hydrolase. And then we have ligases. So what does not ligases, lyase. So a lyase is group elimination by means not using oxygen or water, and they often form double bonds. So essentially, if you see a double bond appear or disappear, it's going to be a lyase. So look at this. Here's fumaric acid, and there's a dehydration that occurs. So what's happening? I'm taking that OH and that H to make the water, right? And I got a double bond there. Okay, so is this a hydrolase? No, you're adding it to the double bond or taking it out. So that makes it a lyase. So here's another one. Here's a double bond, and now it's gone. Water was added to that double bond. So this is an enzyme that's involved in the fatty acid oxidation. So later on, we'll see that this is called beta oxidation. Because remember, here's the carbonyl carbon, that's alpha, that's beta. So here's the carbonyl carbon, there's alpha, there's beta right there. Everything is happening at that beta carbon. And you end up cleaving it right here, and that whole thing goes off as acetyl coenzyme A. And then class 5 are the isomerases. So one isomer is converted into another. No atoms are gained or lost. So remember we talked about people who are galactosemic? They lack enough of this enzyme, galactose epimerase. Epimerase is the old-fashioned word for isomerase. So at carbon number 4, you break the bond and reform it, and galactose gets converted into glucose. They're just diastereomers of each other. And here's another one. 2-phosphoglycerate was turned into 3-phosphoglycerate. So this is done by an enzyme called phosphoglycerol mutase. So a mutase is also an old-fashioned name for an isomerase. And finally, we have ligases. So here we form bonds using ATP or some similar high energy molecule, and these are usually irreversible. So for example, DNA polymerase. When we get to that chapter, I'm going to emphasize that this is a high energy molecule. The monomers are very similar to deoxy ATP. So they're phosphoanhydrides. So when I form a bond linking these together, we get the phosphodiester bond over here, but you're hydrolyzing phosphoanhydride bonds. Later on, this diphosphate right here also gets hydrolyzed. So DNA polymerase polymerizes these bases together at the expense of phosphoanhydride bonds. What about this? Serine and alanine. You put them together into serinoalanine. What does that? The ribosome. And guess what? This is a ligase, but never do they show you ATP being hydrolyzed. When we get to the part when we look at protein synthesis, you'll see that ATP and GTP are absolutely necessary to do this process. The ribosome is a big giant protein, and you grab these things together and you have to push them together to squeeze out that water molecule. How do you push them together to do that? Conformational changes in the protein called the ribosome. How do you do that? Hydrolyzing ATP and GTP. So in the past, enzymes were named for the reactions that helped discover them, often without knowledge of their true biological function. And that's why, you know, people would argue, oh, this is an epimerase. Oh, no, it's a mutase, when they're all the same category. So they formed the Enzyme Commission to straighten all that out. So there's an Enzyme Commission number that describes the chemical reaction catalyzed. So say we look at this one right here. Okay, a common name, creatine kinase. What is its systematic name? Well, it takes ATP and creatine and it transfers the phosphate between them. So here's the EC number. 2 stands for it being a transferase, right? The 7 right there is like, well, what does it transfer? Phosphate. And what does it transfer from? ATP to creatine. See what that means? So it's good that you know what the EC number stands for. You do not have to memorize any of this stuff. I'm never going to ask you, well, what is the EC number for this enzyme? You don't need to know that class 6 are ligases. You just need to know that one of the 6 classes is ligases. Okay. So questions so far? Okay. So levels of enzyme specificity. So complete specificity means absolute stereochemical specificity. The enzyme works on only one single stereoisomer substrate. Most enzymes are that way. Okay. So most enzymes that work with glucose work only on D-glucose. They do not work on L-glucose. They do not work on galactose, for example. So group specificity means that the enzyme works on only a few very closely related substrates. And linkage specificity means that the enzyme works on a variety of substrates that have the same bond. So for example, carboxypeptidase hydrolyzes the C-terminus from any polypeptide. If you were to break down a polypeptide and you had to have complete specificity, you would need 20 different carboxypeptidases. Not only that, you would have to have the genes in your DNA for each of those carboxypeptidases. Later on, we're going to talk about this when we get to DNA and genetics. Why is it that humans don't have that many more genes than, say, an earthworm? It's because the more genes you have, the more things can go wrong. There's a limit to what information that we can handle in our cellular structures. So having linkage specificity for carboxypeptidase makes it we need only one carboxypeptidase instead of like 20. Okay. So let's look at some of the explanations here. So when they were trying to figure things out, I want to say it was before I was born. But anyway, so the first concept was lock and key theory. So I put the word simple in front of it because it is simple. And then the next one I put the word modern in front of it because it's a little bit more modern. But the lock and key theory basically said there's only non-covalent binding between the enzyme and the substrate. The review of chapter 14 would be good. So those are going to be London's, dipolar, hydrogen bonds, hydrophobic, hydrophilic interactions, and salt bridges. And only one key will fit the lock. So within this theory, if the active site is where the substrate interacts with the protein, and it contains binding residues, what does that mean? These are amino acid side chains and catalytic residues. So they're trying to explain why there's so much specificity. So notice that there's a partial negative there and a partial positive there. So that's going to suck this in here and it's going to bind there. So whatever this is could be considered a binding residue, binding R group from the amino acid. And so what if this were an hydrolysis reaction right here? And the hydrolysis reaction in a test tube would be catalyzed by acidic conditions. And I guarantee you, when you suck this in because of the binding residues, that catalytic residue is right there in the perfect position to cause a lower pH in this vicinity right here, catalyzing the acid-induced hydrolysis of that bond right there. Maybe it's a redox reaction. Well, you suck it in there because of the binding residues, and some catalytic residue over here is holding a cobalt right there. That cobalt could be used to reduce or oxidize right there. And so that gives you an idea how it works. But I put the word simple there. I added that. Then later on, Jacob and Monod came up with the induced fit hypothesis. So this is like the lock and key theory with the addition that binding causes a conformational change in both the enzyme and the substrate, which then forces the enzyme substrate complex into the transition state by putting stress on the bonds to be broken. So let's modify this. Here, only one key fits the lock, right? Here, only one key fits the lock. But notice that I made this a little bit bigger than this over here. So the difference is this. The key doesn't need to be pushed into the lock and turned. The key finds its own way to the lock because the enzyme, the lock, has been designed after millions of years to bind to one substrate and one substrate only. So if this substrate were to bounce over here, it basically gets sucked right in there because of the attractive forces between the substrate and the enzyme. See the difference already? The substrate and the enzyme find each other because to keep them apart is a form of potential energy. It's like if I got two water molecules, they're attracted to each other by hydrogen bonds. If I pull them apart, I just created potential energy because they want to go right back together again and form that attraction. So, then here's the other thing that happens. They don't fit perfectly, but they get sucked into one another. That puts a conformational change on the substrate and on the enzyme. They're pushed into each other. And again, what is a conformational change? It's this. Conformational changes can be pretty huge, can't they? So what I'm saying is this substrate binds to this enzyme right here, and it causes conformational changes in the enzyme and on the substrate. So that the bonds that you're going to break in the substrate become stressed already. Now, I admit I've embellished this quite a bit. This stuff right here was never in Jacob Monod's hypothesis. But you understand where the potential energy comes from when they're apart. Not because of the law of conservation of energy. If the potential energy goes down, the only other energy we know about is kinetic energy. So I'm now renaming that kinetic energy as binding energy. What does that mean? That's the energy that drives the conformational changes. So, like all catalysts, enzymes are not changed by the overall reaction. They do not change the position of equilibrium because equilibrium constant is determined by the reactants and the products. It's that difference between reactants and products that determine the equilibrium constant. Rate is determined by the height of the activation barrier. So an enzyme does not change what the reactants are. The enzyme does not change what the products are. It cannot change the difference in energy between them. All it can do is make a reaction go faster to equilibrium by lowering the energy of activation, by lowering the energy in the transition state. So because you didn't have me for chemistry, I don't know what you know, but I'll tell you this, the reactants are stable. They meet the octet rule and the duet rule for bonding. They're electronically stable. Same with the products, right? But in the transition state, where you're transitioning between the reactants and the products, you don't meet the octet rule and the duet rule. Bonds to be broken are halfway broken. Bonds to be formed are halfway formed. You're not meeting those rules for electronic stability. So you have a high potential energy there. But the enzyme or the catalyst comes along and changes the mechanism of how this reaction happens. It changes it so that the activation energy, the transition state, is at lower energy. So it's easier to overcome. So now when we're trying to figure out, what the enzymes do, well, they lower the energy of activation. Well, how do they do that? And you see the words that I underlined? Those are in every single biochemistry textbook. I added this one. It's because all the textbooks copy each other. But I'm trying not to do that. So how does an enzyme lower the activation energy? Okay, so it brings reactants into close proximity of the enzyme. That effectively increases the concentrations at the active site, increasing the rate, because the rate is of this form. Okay. So what does that mean? Going back to this enzyme was designed to bind to this substrate and no other substrate. So that means if they're attracted to each other in either model, you're going to have more substrate around the catalyst than you are out here in the media, because they're attracted to each other by design of the enzyme. And enzymes put the reactants into the correct orientation so that all collisions will be fruitful. Oh, okay, so what does that mean? Here is in the right orientation. Okay. If this thing bounces into the enzyme over here, it basically gets sucked into that pocket right there because of the attractive forces. And let me see if I can give another explanation. Say here's two molecules. What you want is for this reaction to happen. Okay, that's what you want to happen. So look at all these different collisions. None of these collisions are going to be fruitful. But if this gets bound to the enzyme and it's just held like this, then this one gets bound to the enzyme and it gets reorientated and just pushed right into there. That means that every collision now is going to be fruitful. You don't have all this random stuff going on. Then the third one is that it puts strain on the bonds to be broken. That's exactly what we have shown right here. You're putting strains on that bonds to be broken. So if you're looking at the transition state right here, as soon as the reactants bind to the enzyme, those bonds are getting like torqued. So you're not like right here, you're like way up here. So that means it lowers that energy. It makes it easier because just by binding it, you already put stress on those bonds. Then finally, the enzymes provide all the necessary functional groups, either in the form of amino acid side chains or of cofactors. So that's what I mean by this. When this thing does get sucked into the active site, all the catalytic residues you need are right there in the right place to do their job. So that's a lot of concepts right there. Does anybody have any questions about that?
No, too much.
Thanks. So one thing I really like about this part of the course is that when you see that life is a bunch of chemical reactions, and you hear that, oh, they're catalyzed by enzymes, but there's only 6 classes of enzymes, and there is no enzyme that can break any law of chemistry or physics. How is it possible? And I think after this lecture, you begin to see how it's possible. So here's an example. Catalase is an oxidoreductase that catalyzes the heterolytic composition of hydrogen peroxide. Here's what it does, right? So iron is an atom, or I should say is a metal, that causes the hydrolysis. So if you notice, you buy hydrogen peroxide at the store. It's in a plastic bottle. It's a dark bottle because light will activate the electrons in those bonds. But they don't want any metal, especially iron, to come in contact with hydrogen peroxide because it causes this catalysis. You ever put hydrogen peroxide on a wound? Okay, you put the hydrogen peroxide on a wound because it produces oxygen, which kills anaerobic bacteria. He also gives you that bubbling and agitation, which helps clean the wound and also kill any anaerobic bacteria there. But when you put it on your skin, nothing happens. You put it on a cut, it simply just bubbles and goes off. Because the iron that's in your hemoglobin is available to act as a catalyst for this breakdown. Why would your body have the enzyme called catalase to break down hydrogen peroxide? Because you never want hydrogen peroxide to break down in a homolytic cleavage. Heterolytic is when 2 electrons go the same direction. Homolytic is when 2 electrons in a bond go in opposite directions. That gives you 2 molecules of free radicals, right? Hydroxyl radical is extremely dangerous. So what did nature do? This chemistry existed. All nature did to come along is like, hey, I'm gonna take this iron atom and put it in a special protein called catalase, and as soon as hydrogen peroxide comes anywhere near it, it gets sucked into it and pushed up against that iron atom. And I just made that reaction go 1,000, or I should say 1 billion times faster. So in this course, I haven't been concerned about reaction mechanisms. The only reason why I ever give you a reaction mechanism is so you get understanding how it happens, or if it helps you figure out what the products are going to be. Because they're different when a catalyst like an enzyme is done, okay? So let's look at a really complex mechanism. I shouldn't say it's a really complex mechanism. They're all complex. And figure 30. Come on, open up. Okay, so here's some models of human catalase, okay? So you have these monomers, and here's the heme with the iron +3 in it right there. Okay, so here's dimers. These are 2 dimers right here, and they fit together into a tetramer. And then here is one of the monomers over here, And notice that here's serine 95, histine 56, asparagine 129. Here's a water molecule over here. You see what they got this nailed. So here's the heme. Here's how iron is held into it. So the iron in heme has sp3d2 hybridization that gives 6 coordination sites for 6 electron pairs. 4 are used with the nitrogen atoms of the heme, leaving 2 more to react with hydrogen peroxide. Those I couldn't really draw in there because, yeah. I mean, I added all these words and stuff like this. But that's the structure. And where am I on the thing? Oh, here we go. So let's look at a couple of different things. So the first step is that a hydrogen peroxide binds to the coordination sites on the heme iron. When that happens, a water molecule leaves and the remaining oxygen is bound to the active site. So here's the enzyme, here's its iron, here's the first hydrogen peroxide. So a water molecule leaves, and now this radical right here is bound to the enzyme. Okay, that's the transition state. So here I drew it over again. And now the second hydrogen peroxide comes in, and another water molecule is given off, and also an oxygen. And I regenerated the catalase, right, which is the enzyme. So let's look in depth at the role of the heme iron. So the heme is symbolized as boxes. Holding the iron is iron plus 3. So here's the heme, right? So because of the sp3d2 hybridization, there's 6 coordination sites on the iron. They can accept electron pairs. 4 are used by the heme nitrogens, leaving 2 more to accept electron pairs on the hydrogen peroxide. So the sp3d2 is shown over here, okay? So here's the S, here's 3 P's, here's 2 D's, okay? So this is the electron configuration of iron plus 3, okay? So then the iron plus 3 pulls electron clouds from the hydrogen peroxide into one of the 6 coordination sites, okay? So here's histidine, here's the hydrogen peroxide right here. So the oxygen gets pulled in over here and temporarily forms this double bonded oxygen over here. But it's got an extra electron, plus it also has a positive charge. This is the transition state. This is extremely reactive, isn't it? Notice that the histidine right here is totally involved in doing this, okay? So is the arginine over here. So the arginine is providing positive charge that attracts this partial negative charge over here. So these are the bonds that's going to be formed. This is the bond that's going to be, this is the bond that's going to be broken right here. And then you form this double bond over here. So here's another view of step 1, showing you the release of water. So here's the heme. Here's the histidine again. Here's the asparagine. See these attractive forces right there, holding it just right in the right place? And then you form this double bond with oxygen right here. If you look at the other parts of the protein involved in the catalytic site, they're all involved in this. So the non-binding electron pair on the basic side chain of the histidine attracts and pulls hydrogen from the hydrogen peroxide. The arginine basic side is positively charged and stabilizes the partial negative formed on the hydrogen peroxide in the transition state. And then the iron plus 3 temporarily holds on to an oxygen and becomes this radical right here. And it simply waits for the next hydrogen peroxide to come in. Okay. So here's another view of step 2 showing the release of oxygen in water. So here's the transition state. The next one comes in, and oh, here's oxygen, and there's the water right there that can now leave. And you regenerate the asparagine, you regenerate the histidine, and all this stuff goes back to normal. Wouldn't this be a great essay question for exam 3? In 5,000 words or less, explain the role of the heme iron. Yeah. So it's in a red box, it's not on the exam. I just want you to see one example of an in-depth reaction mechanism catalyzed by an enzyme. It also shows you just how much we really know. So an enzyme is a stereochemical catalyst. It's specialized to do one thing and do one thing really well. And so notice, I broke this up into 2 steps, but actually there's many, many more steps in here that weren't really emphasized. But here's another way. If you did it in a test tube, you'd have to deal with this. Everything has to be done at once. When you break it up into smaller steps right there, each transition state is so small, it's easily done. So hopefully that makes sense. So let's see, 12:15. Yeah, so we're about out of time here. So anybody have any questions before we quit?
So the iron heme example is kind of showing the modern induced fit hypothesis.
No, it doesn't really show the induced fit, this part in the red box. But here's what we mean by induced fit. When this comes in here, notice that there's an attraction between here and here that wasn't necessarily there before. So those attractions pull it close to each other, causing a conformational change. That is the induced fit. But this really here is more of the electron thing. We're not talking about in a general sense the induced fit. It's just when you get a conformational change. But notice that when you got something big like this, I don't have to do a big giant change like this to affect some bond in here. Because if I got a little bit of rotation here, a little bit of rotation here, and a little bit of rotation here, all those rotations are easy to do. But it can result in a large rotation in one specific part of the molecule. And really, this is the reason why enzymes are so big. They're so big compared to the subject that they work on, because you take only little conformational changes throughout that large structure can cause a large conformational change in the active site. I'm not sure I answered your question, but
No, that helps. So like tiny, like small changes impact the overall confirmation of change that occurs.
Yes, it does. Yeah.
Okay, I see.
Yeah. So in other words, when you look at this, does this histidine have to like reach way over and pull that, and that heme really reach over? And no, they're kind of pushed together when this thing binds because of the attractive forces that are shown there. Yeah. But you're right, it's hard to concentrate on everything all at once.
Yeah, definitely.
Yeah. Well, just think how hard it is for the enzyme. Any other questions before we stop for today? Okay, well, don't forget you have your exam to do if you haven't done it already. And I'll send an email for when our next lecture is tomorrow, probably at noon. Okay.
I got a question.
Does somebody have a question? I heard something.
You know, I'll just text it. I'll just send you a message.
Will you send a message?
I'll just email you.
Okay, sure, fine.
Okay, thank you.
Okay, you folks have a good day.
Bye.
Take care.
Thank
Thank
you.
you.
Bye.
Bye.
Bye.